This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 168, recorded January 27th, 2012. Hey everybody, I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today here in TWiV studio, Dickson Despommier.
Hello, Vincent.
D-cubed.
D-cubed.
Or 3D.
3D.
How you doing?
I'm doing great.
Yeah, you sound distant.
No, I feel fine. I'm about 5 feet away. Maybe that's why.
Good to see you again.
Good to see you.
I saw you all week. I don't have to pretend.
Of course not.
Also joining us from North Central Florida, where in just a few days there will be a presidential primary, Rich Condit.
Hi, fellas.
Hey, Rich.
How you doing?
How you, Newt?
You calling me Newt?
He turned me into a newt. You remember that line from Monty Python?
No, I would never call you a Newt.
Good, thank you.
You got tenured. He didn't. Dickson is holding up a newt.
That's true.
Is that a newt?
It is. It's a black-spotted newt.
Cool. There you go. How you doing, Rich?
Great.
Is the weather really nice today?
Beautiful blue sky, 71 degrees.
Oh, it's really awful here.
Yeah, we got a miserable. But it's warm.
It's been pouring.
It's abnormally warm.
We have two additional participants from the state of Massachusetts. Out in the western part, Rich Condit.
No. I got all that wrong.
Joining us from western Massachusetts, Alan Dove.
And what a state I'm in.
Hey, Rich, I didn't know you moved so quickly.
He moves at the speed of light.
How are you, Alan?
I'm doing well. I actually interviewed Newt Gingrich once.
You did?
Yeah.
What'd you
for
ask
what
him?
purpose?
I talked to him about science policy. It was for the New York Academy of Sciences.
Yes.
Their magazine.
And he said?
It was about science education, and he had some actual intelligent thoughts on the matter, and interesting guy to talk to. I don't think I'd vote for him, but he was an interesting guy to talk to.
Uh-huh.
How long ago was that?
That was about a year and a half ago, 2 years ago.
Oh, not that long ago, huh?
No, not long ago.
Curious. You must have lousy weather up there as well, right?
Oh, yeah, it sucks.
It's been raining all night. It's cloudy right now.
It's pouring.
It's terrible. Let me tell you what the temperature is here. I'll get up
It's 60. I bet you it's 60 degrees.
60 degrees Fahrenheit. Are you serious?
It's supposed to get up near to that.
Currently in New York City, it's 11 degrees. 14 degrees centigrade. Wow, it's quite warm. Also joining us from the state of Massachusetts, third time back, I think, from the city of Boston, Welkin Johnson.
Hello, and happy to be back.
Yeah, welcome back, Welkin. I just can't resist. Do people say that all the time?
I've never heard that before.
Oh, really?
No. I think I've heard it on this show before.
We have.
Yes, I think so.
Now, since we last spoke, you've now moved to a different university, right? You're at Boston College.
That's right. That's right.
Right in the city of Boston, is that
Not
correct?
a Jesuit school?
Yeah, we're right on the sort of the edge. We're on the edge of Brookline and Newton, so it's the Chestnut Hill campus. And I guess I'm not fully moved in yet. I have the equivalent of the Berlin Wall down the middle of my lab right now.
In boxes?
They have a big a temp wall up because they're doing renovations on the lab to get it ready, and so we have a big picture of Checkpoint Charlie up on the wall.
Just the thought of moving a lab makes me sick to my stomach.
I did that 4 times.
Oh my God! I never want to do it again.
No, I don't blame you.
No,
I don't blame
I
you.
did it once. I'll never do it well. Obviously, now I'll never do it again. But I swore then I would never do it
And
again.
they never do it well. They don't do it well. They don't. They try, but it just doesn't work out usually.
So where you are is not the medical school, is that correct? Or it is? School of Medicine or no?
So BC does not have a school of medicine.
There you go.
So BU has a school of medicine. BC has a law school that's pretty well known. So there's the main campus and the law school.
Right. So are you actually going to have to teach for a living?
I have. Yeah. I have. Yeah, actually, I've already started teaching.
Cool.
So it'll be the same job, but a lot more undergrads sort of wandering in and out of the office.
Right.
You're a professor of biology.
That's the other thing I have to get used to is all these people keep calling me Professor Johnson, and I don't respond.
Yeah, I know that. It doesn't happen at medical schools. That's
You've
for sure.
never moved, is that right?
No, I came here to Columbia in '82. It was my first job, and I've been here ever since. Yeah, we've moved within the building, but not without. Nope. It's kind of nice, and I think this will be it. I'm not going anywhere else until I'm finished. That's it.
You know, I stayed here too. I've been here since 1971, but I've moved 4 times in that time.
Yeah.
And it's very disruptive. It's extremely disruptive.
I think the record goes to Jeremy Lubin,
Yeah.
who's moved between continents multiple
That's
times.
true. That's true.
Yes, that is quite ridiculous. But he's coming back to Massachusetts.
Yep.
Maybe we'll get him on one of these days, although Switzerland does not permit him to leave yet. They said you have to give a year's notice, and
Really?
he didn't realize that, and they said they would put him in jail if he left before a year.
Leave it
Wow.
to the Swiss. That's
Really?
great.
Although I don't know if you leave, how they can put you in jail if you're the U.S. Certainly we would not extradite him, so he should just leave.
They'd freeze his bank account.
Yeah, you're right. I'm sure it's full.
His numbered bank account.
All right, so we have 2 cool papers today, and the first one has been actually requested by a number of listeners. Came out a bit ago in January. No, it's just published in the paper version, but it came out online, I think, in November. It is from the laboratory of David Baltimore, TWiV 100, antibody-based protection against HIV infection by vectored immunoprophylaxis, VIP, another acronym. I actually picked this because you were going to be here, Welkin.
Really?
Thought you might like it. Do you like it, or should we throw it out and do something else?
No,
Well,
I like
nothing
it.
interesting
It's-
ever comes out of this lab anyway.
That's right. I should have asked you, but I figured anything retroviral would be good.
David Baltimore, I've heard of him.
Right.
He was on TWiV.
Yeah.
He was.
Number 100.
It was
For
fun.
the hundreds. That's
was
right.
fun.
It made him famous.
Yeah, right. Hey, Rich, what made him famous?
Reverse transcriptase.
That's right.
There you go.
Nobel Prize for that guy.
Betcha.
He and Howard Temin.
And he hasn't exactly slacked off since then.
No, I was going to say he has a consistent record of being a pretty smart fella.
Yeah, actually, if you look at the biographies of a lot of Nobel laureates, they either get the prize at the very end of their career, or they do one thing and then
Flash in the pan, right?
So it's a flash in the pan. But he's just one of the most consistently productive scientists ever, I think.
That's right.
Well, look where he was from.
Where was he from?
He was at Rockefeller.
Yeah, he got his PhD there. That's right. Yeah, he was here last. Well, I guess that's when we did 100, and actually he talked a little bit about this stuff during his seminar. I mean, the basic idea is that it's about HIV, of course, and we haven't been very good at developing a vaccine, so he says, why don't we give antibodies? We have some neutralizing antibodies that have been identified. They work. Well, why not deliver those?
Now,
Right, the problem is, how do you deliver those?
I don't quite understand these
Intramuscularly.
antibodies because we make antibodies to HIV, right? But the problem is that we can't keep up with the virus because it mutates. Are these to like conserved epitopes or something like that? Where do these antibodies come from?
Right. My impression is there have been several of these antibodies that have been isolated that are these broadly neutralizing antibodies from a few cases. And in the paper, of course, they talk about a few different ones and they try a few different ones. But I assume that it's just very conserved epitopes on the surface of the virus, right?
Yeah. I think there's 2 parts to it. It's antibodies to conserved epitopes, but it's prophylaxis.
Right.
So I think the idea is if you have the antibodies there ahead of
Start
time.
with. Right.
Yeah.
Right, because I gather
Before the
some of
virus
these
gets
antibodies arose in people who it's too late for
Yeah.
that fix
Yeah.
the problem.
Okay.
But if you can limit the virus, then its chances of escaping from the antibody will be a lot more
Yeah.
difficult.
Okay. So and so the idea here, obviously, is not obviously. The idea here is to give these people this antibody before they've been infected. So it's a kind of immunization.
Right.
It's an active passive immunization. That's interesting.
Right. And I think
point.
this is distinct from the elite controllers, right?
Yeah.
So the elite controllers are these couple of people in the world who've managed to develop an effective immune response against HIV after infection.
Yeah, and that's mostly cellular, I think, as I recall.
Whereas these antibodies have arisen by somatic hypermutation. So the people got infected, and over time their immune systems refined, and they finally got some antibodies that are against conserved epitopes and neutralize the virus, but the virus is already in cells and doesn't have to be exposed to the antibodies anymore, so it doesn't help those people. But if they'd had those antibodies, if you could go back in time and give them those antibodies before they got infected, then it would be helpful. Maybe.
Yeah. And I think the other I don't know if they go into this, but the other background to this is a number of these antibodies have been shown, proof of principle has been shown because they work by passive transfusion in animal models.
Okay.
Right. So not delivered as a vector, but by giving monkeys a bolus of these, they've been effective.
Right.
The problem though, is that the antibody doesn't stick around forever.
Right. So there you inject the antibody, and then you turn right around and you inject the virus, and you find that the monkey's protected.
Right.
But you can't just go injecting everybody with the antibodies every day.
No.
So I was interested to note that these antibodies are extensively somatically mutated because they've gone through many rounds of selection in B cells. And I thought everyone would like to be referred back to Concerto in B.
Yes.
With that whole process of selection in the lymph nodes we talked about with Gabriel Victora.
Yeah, that actually means something to me now.
Yeah. There you go. So it's also a characteristic of some of the broadly neutralizing influenza antibodies that have been described, which are also directed against the highly conserved sequences of the HA. They're also extensively hypermutated.
Right, which could be useful for flu if you get a related strain later.
For flu, for sure.
But in the case of HIV, where you've got a chronic infection going on, this is just too late for the person who's infected.
In my class yesterday, we were talking about these, and someone said, well, if we had those antibodies and we could figure out how to use them, then we wouldn't worry about H5N1, would we? I said, yeah. If we
it's
had
paper
it.
number 2.
We don't have it.
Maybe another important point for the for listeners is to realize that just because you have antibodies, they're not practical because they're very hard to make in bulk.
Yeah.
Right? It's not like a drug or something that you can give somebody every day. Making enough antibody to give to every patient every day would be probably impossible.
Well, there is a therapy, an antibody therapy for respiratory syncytial virus. This is for babies who get infected with this virus, a very serious illness. And there's a monoclonal antibody produced, but it is incredibly expensive.
Yeah.
Thousands and thousands of dollars for a course of therapy. So it's not really practical.
Right, and it's only useful in acute cases like that, where you're going to give a large dose, and you need something that's going to act in 24 hours.
Yeah.
So for an HIV vaccine, you need something that's going to protect somebody for 30 years, and that's not going to be done with injecting
I have
antibody.
a question. Back in the dark ages of immunology, which is when I trained, we talked about idiotypic antibodies and then anti-idiotypic antibodies. And the anti-idiotyp was the mirror image of the pocket of the combining site for the idiotypic antibody. So here you have an antibody that protects. And I know that this antibody will elicit anti-idiotypic antibodies. So that's the actual antigen that you're aiming this antibody against. So couldn't you find the anti-idiotypic antibody against this antibody and then deduce the antigen from that?
Can you say that all again?
Sure. It's like making a mold, and then you squirt plastic in it, and then the plastic sets up to reflect the exact combining site between the light and the heavy chains. So you've got this little plastic thing that you can pull out and look at, and then you can say, oh, that's what the antigen looks like.
I think that the Scripps group, Dennis Burton and those guys, I think they have done things like that where they will pan these antibodies against like peptide libraries.
Yeah, exactly.
To identify peptides that can function as a mimic for the real antigen.
Right, so one strategy for possibly developing a vaccine would be to try to find the antigen that would elicit these antibodies and then just give the antigen vaccine.
Exactly.
It's It's a very interesting point, though, in with these antibodies, in particular the HIV antibodies. A lot of people have spent a lot of time trying to develop artificial immunogens, and what happens is a lot of times you will get ones that will induce antibody, but that antibody does not work against the virus. And what it means is that the artificial antigen, that there's more to the story, that the actual thing that's being recognized is much more complex in the context of the virus.
Right, and because these are arising through somatic hypermutation, it suggests that what's actually responding to on the virus is something that's not in and of itself very immunogenic.
Yeah, that's right.
Because if it were, then you'd see these antibodies showing up in people just as they got infected, but you don't. It's after they get infected and their immune system's been working on it for a while, then they do somatic hypermutation, and they end up hitting on these neutralizing antibodies.
So this research also suggests that there's a threshold beyond which, if you exceed that with viral production, the antibodies don't work so well. So this is a preventative rather than a curative.
Yes, this would be a vaccine that's focusing on developing these neutralizing antibodies would be, I would think, strictly prophylactic.
Right, head them off.
Because we know that there are people walking around who have these antibodies who are infected, and it's not curing their virus.
Sure. I mean, that's the same thinking process that goes into the malaria vaccine.
Well, whether it would be or not would have to be tested, right, Dickson?
Yeah, of course.
As we'll see, it works great in mice. But
no, absolutely.
Ferrets are not people, and mice are not people either.
Neither are corporations. I think Newt might want to know that one.
The approach here was to clone the genes encoding these antibodies into an adeno-associated virus vector. Have we ever mentioned AAV on TWiV?
I don't know. I don't recall that we have. We must have mentioned it, but we've never done a big shtick on it.
I think I think exactly that. We've mentioned it maybe in a gene therapy context or something, but just kind of glossed over it.
I suspect Rich knows all about it because one of your colleagues down there at Gainesville, Nick Muziczka, right? Muziczka.
It takes practice.
He
Muzyczka. Muziczka.
He works on this guy.
It takes years to become a Muziczka, and yes.
And he's another guy who's moved around a lot.
Muziczka actually is primarily responsible for developing AAV as a gene therapy vector.
Right.
He's on the patents, etc. And yes, he did that in his first tour of duty at University of Florida when Ken Burns was the chair of the micro department that I'm in. And Ken actually brought AAV and Nick, if I understand it correctly, to UF and spent a number of years here during which this was developed. And then Nick went to Stony Brook for several years, and Ken went elsewhere as well. And then they both, in separate moves, came back. Ken as dean and Nick as the director of the new, what is now the Powell Gene Therapy Center. And so there's a pretty robust gene therapy program here and a lot of vector development. And so this is sort of one of the centers of AAV as a gene therapy vector.
So can you give us the executive summary?
AAV, adeno-associated virus, is a parvovirus. So we're talking really small, 5 kilobases, single-stranded DNA in a naked capsid. And it is a human virus that, as far as anybody can tell, is entirely non-pathogenic. It's just hanging out. There is one identified pathogenic parvovirus, so same family in humans, and that's B19, causes slap cheek syndrome. What is that?
Fifth disease.
Is Fifth's disease, right? It's not roseola, it's Fifth disease. Fifth disease. It's a mild skin rash that most kids get. At any rate, AAV apparently doesn't do anything. It's called adeno-associated virus because it's such a lame virus that it needs help from another virus to even replicate. All right? It needs adenovirus to co-infect cells and kick the cells into a cell cycle to provide the replication proteins that AAV needs to replicate. And interestingly, so it's a single-stranded DNA virus, and when the DNA enters the cells convert that to a double-stranded molecule, and that initiates the replication cycle. And towards the end, there's a magical AAV replication protein that's responsible for helping spew out single strands to be packaged in this small capsid and get out. So if adenovirus isn't around, interestingly, this thing will actually integrate into the human genome, and it does so in a highly site-specific fashion. There's a unique site on chromosome 19 that this thing integrates in, and it sits there in a quiescent form. And if you take cells where that's integrated and then with nothing else around, and then superinfect them with adenovirus, then the AAV will replicate out and go through an active replication cycle. But I think the fact that it was non-pathogenic and the fact that it had a unique replication site were two of the factors that inspired people to develop it as a gene therapy vector to start with. Because the thinking was, first of all, it's non-pathogenic. That's good. Secondly, you could get it integrated into a specific site rather than random integration that might be mutagenic, and so you could get long-term expression of a transgene from a known site. So I think that inspired some of the first development. Now, as it turns out, so what you do is you gut this thing. You basically take out both the capsid and the replication proteins, leaving only the terminal parts of this genome that are responsible for the replication and packaging. And you can then replace the guts with your favorite gene driven by whatever appropriate promoter there is. And you can take plasmids that have that construct on it, introduce them into cultured cells, and also introduce plasmids that have the replication and capsid proteins, and you can get out of that the recombinant DNA that's encapsulated in this capsid, and then you can purify that and inject it into a patient, and it will deliver this DNA, which will then express the transgene. And I think one of the initial hopes was that this would then go to the chromosome and integrate and give you permanent expression. It turns out that when you take out the rep protein, in addition to the capsid proteins, it doesn't integrate anymore. However, fortuitously, it turns out that it establishes itself as an episome, that is a circular piece of DNA separate from the chromosome, and will stay there and express genes indefinitely. So it's almost even better than the integrating guy, because it will give you very, very long-term expression without integrating at all. So it turned out to be okay after all. The disadvantage of this thing is that there's a limit as to how much DNA you can put in it, because the genome's only 5 kilobases of single-stranded DNA, so you can only put in about that much, otherwise the capsid won't hold it. And they talk about that there are newer versions of this that incorporate a double-stranded DNA rather than a single-stranded DNA, which makes things more efficient.
How often are people exposed to this? Is it carried by animals?
Everybody's got it.
So we have
AAVs
pre-existing.
Yeah, basically, it's very prevalent. Almost everybody's got AAV. You don't have it?
Just kidding.
So actually, I think one of the concerns about it initially as a gene therapy vector was everybody's got antibodies against it, but it turns out.
And in fact, that has been a problem. Lots of time in some of there's also a ton of different serotypes, and there's a lot of research going on in engineering the capsid to target different tissues, and also to overcome this problem of mounting an antibody response to it. And they've been pretty successful with that now. It's there are among these different serotypes, and in fact hybrid serotypes, you can manipulate the thing to have a preference for one tissue or another, and also get around the immune response that might otherwise compromise the use of it. So it's grown to be a pretty sophisticated vector.
I was interested in the modification that they used here, which I had not been aware of, and that is normally these vectors have single-stranded DNA in the virion, and getting from single to double-strand is problematic. It reduces the efficiency. So if you make it double-stranded to begin with, you get a much more efficient transduction, right?
Right, and they call that the self-complementary because it's actually a dimer of the genome that's self-complementary, but in effect it's double-stranded. You pay a price for that because, as I said, the limit on cloning into this thing is the size of the capsid. Yeah. So if you're sticking in double-stranded DNA rather than single-stranded DNA, you can't stuff in as much, and so the amount of transgene that you can stick into it is less.
Nice.
But at any rate,
cool.
They succeeded with this thing.
Right, but there's enough space to cram in, well, as we're going to talk about, an antibody.
As a naive person sitting here listening to all this, why can't you just get a bigger capsid?
Yeah, you can't. That's...
Would be a different
virus.
They didn't
think of that.
It'd be a different virus, that's right.
Just empty out another capsid and use that. What's wrong with that?
This capsid binds this genome,
Right.
Use another capsid, it's not going to recognize.
Highly adapted to this particular genome.
Hey, you have to use a different virus, Dickson.
Well, of course that's the answer, but
this has got great qualities, as you've heard, right?
Of course.
Right. Well, so in order to get this genome packaged, you need this unfortunately small.
I mean, antibodies are heavy and light chains, right?
Yes.
Yeah, I was just. Oh, I see. So the expression vector. I'm looking at this in the supplemental data.
Right.
The expression vector expresses both of those.
Yeah.
The heavy chain and the light chain.
Yeah. You know how they do that? It's very interesting. They put the heavy chain and
they
then
got the 2A protease in there.
It's not the whole protease though. It's just a part of it. It's a part. It's actually not a protease. It's a 2A sequence from certain picornaviruses that causes pausing and reinitiation.
Oh.
So you get two proteins made, but there's no cleavage necessary. You get termination and reinitiation. It's called the self-processing sequence, but no one really knows how it works.
And the disulfides line up by themselves?
The disulfides in the antibody?
Yeah.
Sure.
Okay.
That's all in the
self-assembly.
Yeah.
Yeah. Okay.
Yeah, so they extensively rebuilt this vector. In fact, I think that's a big part of what they accomplished here was they tinkered with this vector until they got expression levels that they liked. And they rebuilt the promoter, and they've got this 2A sequence in there and a bunch of other. So there's the supplementary data, which, of course, like most papers these days, that's where most of the results actually are. They've got this whole description of the rigmarole they went through to get this vector working the way they wanted it to.
It's just incredible. These things are so highly engineered now with little bits and pieces from all over the place. It's unbelievable.
Yeah.
So they engineer antibody into the AAV vector. They infect mice, and they make a ton, 20 to 250 micrograms per mil in the serum, and they just make it for as long as the mice live, right?
Yeah, they stick it into the muscle, into the gastric nemus, which is the calf muscle.
Sure.
Right, so they just give them an IM injection of this thing.
It's amazing. One injection.
And it'll stay there and just keep churning out antibody.
Right, 64 weeks they followed this, I think.
I bet they die at week 65.
Yeah. How long,
no, just kidding.
How long does a mouse live?
Not very much longer.
A
couple of years at most.
Yeah, a couple of years.
That's right.
Nobody knows.
I don't know if there was any pathology. They don't say anything. I mean, nothing overt, but gee, that's a lot of antibody, isn't it?
Yeah, it's huge.
Yeah.
So, I mean,
well, they comment it's 100-fold above a protective antibody level if you were to extrapolate this to humans for typical antibody response.
The interesting thing is they get really minimal or no response against the antibodies, right?
Right.
Right.
These mice are also not just, they didn't just catch these off the field.
Right.
Yeah, they're immunodeficient mice.
Immunodeficient.
But they also injected some immunocompetent mice.
Right.
And they said very limited antibodies against the antibody in Black 6 mice. The BALB-Cs made a little bit, but it didn't appear to impact levels of the antibodies, the IgGs. So it's interesting. I don't know why it wouldn't be immunogenic. But, people will be different. Anyway, the immunocompromised mice or deficient mice, then they engraft those with human peripheral blood cells and then reconstitute human immune systems, I guess. Is that how it works, Welkin?
Yeah, that's right. That's the basic idea. So I guess that's a test of whether or not these antibodies are being seen as self by a human immune system or not being seen, which is what you want.
Yeah, yeah.
It's probably the only organism on Earth that could answer yes to the following question: are you a man or a mouse?
So having a human immune system allows them to infect them with HIV. So that's a model, I guess, for HIV infection, because they have CD4 cells. Mm-hmm. And you look at depletion of those, right?
Yeah.
And these guys are protected.
Mm-hmm.
Yeah.
And they give them a ton of virus.
Right. So the control mice that they injected with this vector that's just expressing luciferase, reporter gene, their CD4 cells plummet.
Right.
And then the ones that they gave the antibody expressing virus, they're fine as far as you can tell.
I'm looking for how much virus they put in. Does anybody remember?
They give it in.
Loads.
They even have a dose response down here, and they don't talk about it in PFUs. They talk about it in
milligrams,
nanograms, which doesn't make a lot of sense. To me.
125 nanograms. I bet that's a lot. Well, it doesn't plaque this thing, right? This virus.
No, but I mean, at any rate,
yeah, it's a lot of virus.
It's a lot of virus.
It's a lot of virus.
And so they're totally protected. It's amazing. They also use they use a couple of different antibodies. They have one here called VRC1, which neutralizes over 90% of circulating HIV strains. And that works too. So
they have a couple of other antibodies that they try that are not as good as their original B12. So, different antibodies work differently.
Now, Welkin, you had said this had been done previously with SIV.
Yeah. So reference 14 in the paper is a publication from Phil Johnson and Ron DeRozier where it was the same idea, except they used the SIV model in monkeys, and they used, actually instead of full antibody, they engineered the AAV to express single chain FV, which is a sort of a truncated, it's just the business ends of the antibody, I guess, and it worked. It worked fairly well. It did raise the issue of whether or not you get immune responses to the antibody itself, right? Because those antibodies had not been engineered to look like monkey proteins. Mm-hmm.
Mm-hmm. Right. These are basically human antibodies. So presumably in a human trial, humans would tolerate them quite well in that regard.
Yeah. But the paper's worth mentioning too, because I noticed Baltimore, when he talks about this study, he's very quick to point out that mice are not men. Mm-hmm. Right? And the nice thing about the precedent in the monkeys is that monkeys they're not men either, but they're a lot closer than mice.
That's true.
Right. So it suggests that if these guys have worked out all the details, you put that together with the previous work in the monkeys, and it suggests that this actually might be feasible.
Yeah.
Wow.
So the DeRozier study was done 2009, right?
I think that's right.
So why hasn't this gone into people since then? Because that was SIV, I suppose, right?
Yeah, that was sort of an ideal conditions sort of experiment. It was an antibody-sensitive SIV and a known antibody that worked against it.
Mm-hmm. Okay.
So it seems to me that ideally, in order to be completely successful, this would have to be good enough so that it prevented the virus from ever establishing infection. Mm-hmm. Right, because if you got virus integrated and maybe could tick over a little bit, it might be able to evolve to get well, I don't know. I'm seeing a race going on in my mind. I guess I'm asking, how are the monkeys doing?
Right.
The monkeys are still clean?
I don't know what the latest is on them, but I think there's really two parts. There's two levels to what you're bringing up, right? There's the level at which to actually protect an individual, right? You probably would want to prevent infection completely. But on a larger scale, you think about a treatment like this, if it just reduces the probability of getting infected.
Right.
And let's say you have to be exposed a lot more, then you could put a dent in the spread of the virus in a region where there was a lot of endemic HIV.
Right.
Yeah, it's going to, if you don't have a 100% effective vaccine, but you have a 90% effective vaccine, you're still in the game.
Right.
And then the question is, well, how far down could it go and still be useful? And I think we're seeing some of that with the malaria vaccine development. It's not It's not nothing, but is it good enough?
It's important to remember with a lot of this kind of the stuff that's going on, gene therapy, vector-based approaches to trying to treat HIV-1, that the most likely place that they're going to be used is in places where the virus is a very serious problem or where there's people who have no choice, they're going to be exposed to the virus.
Right.
So even if this worked, it's very unlikely you would be vaccinating children in the United States with it, for example.
Right.
It wouldn't be applied because there's other ways to avoid getting HIV-1.
Absolutely.
So there's no barrier to AAV, right, Rich? Because those have been used in human clinical trials already.
That's correct.
But none have been licensed yet, right?
I do not know that.
Mm-hmm.
Okay. I think one of the most advanced therapies is therapy for retinitis pigmentosa, a couple of different eye diseases.
Right. So the real issue here is whether this amount of antibody would be a problem in people, I guess, and you'd have to go through a lot of animal work first before you did your phase 1 to assess that.
Right, but too much antibody response is kind of a nice problem to have.
Yeah, I'm just worried about immune complexes and
but if you get this vector into a primate model and it's still 100-fold more antibody, the super prophylactic response that they talk about, and the monkeys seem to be getting sick from it, then you could maybe tone down some of the rigging they did on the vector. The promoter, well, they picked a high-activity promoter. You could go with lower-activity promoter, and you could play around with that and try and come up with something that...
So, Welkin, does this have to go into a primate model next?
You mean before it would go into humans?
Mm-hmm.
I'm actually not an expert on that kind of thing. I don't know whether that would be required or not.
If it's the FDA looking at it, they're going to want to see this in a primate, I'm sure.
Yeah, it seems like a good idea. I don't know.
They're certainly going to want to see it in something besides a mouse.
The early antivirals didn't have to go into animals because there were none at the time.
Right.
But they fast-tracked them from cell culture results right into phase 1.
That's true.
I suppose with something, I'm just thinking out loud, but with something like this, that the bits, all the pieces of this taken separately are things that have been through trials in humans or used in humans in different ways. So they might be able to argue that there's nothing about the initial treatment that needs to be studied in a monkey because it's already been done, maybe.
So, Rich,
antibodies have been done, passive transfer has been done, AAV's been done.
So, Rich, you remember with respect to these smallpox antivirals, is the 2-animal model rule, right?
Right.
And so that would argue that you have to put it in a primate model.
Well, you'd have to go into some. I think anybody's going to want to see some other animal model, so it's not just mouse to human.
Well, you do a bunch of animal safety tests, right? Not necessarily protective types of assays.
Yeah, test for toxicity. Yeah. Well, in fact, that would be a good primate study to do, is whether you get protection and whether you have a good HIV model in primates, which of course is problematic. But you could inject a bunch of monkeys with this and see if they get sick.
I don't know. I thought this was really neat.
Yeah.
I mean,
it's awesome.
The tech is great. It works. I think it's very promising. And frankly, I'm surprised. I would never do this myself, express antibodies in people. I would just think it'd be so problematic, but just goes to show you never know who's going to do the experiment and get it right.
You just have to try sometimes.
And this is supported by the Gates Foundation, we should add. So it's good use of that kind of money. Grand Challenges in Global Health.
Yeah.
It's at David Baltimore.
So I have another question then, which relates to the level of IgG that circulates. Again, back to the dark ages of immunology, we were told, at least let's say in the 70s and 80s, maybe even the early 90s, that there's an upper limit to the concentration of IgG. So if you're at that level already with this high expression level, are you going to inhibit the induction of antibodies to other things?
Good question.
That would be something that you could look for in the animal model, though.
Yeah,
correct.
I bet they're doing it in the mice, right?
Because there's a feedback mechanism, which I was told about at least, that still probably operates now, even though we're in 2012. That there's a way of shutting off antibody synthesis as well by not just anti-idiotype, idiotype, anti-idiotype, anti-anti. It just goes on and on, like looking in the mirror in a barbershop. That the concentration level matters as well. So
you go to barbershops, Dickson?
in the old days, I went to barbershops. In the dark ages of my hair, when I had hair, I went to the barbershop.
Well, and Rich sings in them, right?
That's right.
Anyway, I like this a lot. It's a good paper.
Oh,
yeah, very cool.
the tech is amazing. There's so much work in this. I've been doing this for a long time.
While you were describing it, I couldn't help but think also that there are many reality shows on television, and two of them, one is about making motorcycles from scratch, and the other is about hot rodding and taking a normal car and making it look different. And having these guys, if they hadn't been virologists, they probably would have been hot rodders. Because they're just redesigning everything to suit their own purposes, and it's just it's a marvelous mindset. I
gene
totally
jockeys,
agree.
man, they're gene jockeys.
Gene jockeys, and I say
CalTech made a video about this. We'll put a link to it.
It's good stuff.
It's really
very
slick.
Good stuff. I think the mindset is the same.
Before we go on to our next paper, I want to ask Welkin. So you've heard me in my lectures tell students that retroviral RNAs are plus-stranded, but they don't get translated. So what's the reason? Because they always ask me that.
Yeah, so this came up because I just made my students listen to TWiV 43, I think.
Uh-oh.
There's a couple in the early 40s where you do the neat trick of telling the students how, just remember that every virus has to get to messenger RNA somehow.
Mm-hmm. Yeah,
right? And so if you know the genome of the
101.
virus, then you can you can sort of figure out the steps that it has to take. And I think you were explaining it to Dick on the episode
That's
that
true.
They listened to.
This is the equivalent to the Idiot Abroad type show that's on now. I'm the idiot type.
And so there's two explanations, I think, that I can think of for why the retrovirus mRNA is not used, or the retrovirus genome, even though it's a positive-stranded and capped polyadenylated RNA, is not used as a messenger RNA. There's the mechanistic explanation is that unlike the other RNA-dependent polymerases, reverse transcriptase actually has two activities, right? It's a polymerase, but it's also an RNase. Mm-hmm. And that's the mechanistic explanation. It destroys the genome. But from the point of view of students trying to be able to work backwards and figure this out, I think that the simple thing that has to be remembered is that reverse transcriptase actually has to make two things, right? So it takes that RNA strand, it makes a single-stranded DNA, but it doesn't then unwind those and let them go floating away. It has to hang on to the single-stranded DNA and make another strand of DNA. Mm-hmm. And so it has to get the RNA out of the way.
So I guess you could say, why don't the ribosomes compete with the RT then?
Yeah, so the RT never lets go of it. I mean, I guess the other explanation is it's always bound up inside of the capsid complex.
Yeah, right. All right. Yeah, we just talked about this in class the other day.
Right. So the RT is already on the scene when the genome arrives.
Yeah, it's all wrapped up.
Yeah. And it's it just starts right to work, and then there's no positive sense RNA when it's done.
Right.
Ribosomes never get a chance.
The podcasts work great for the class, by the way.
Oh, yeah?
Oh, good.
Yeah. I bore them for an hour, and then they can go listen to you guys.
Well, now they
and
can
we bore them for an hour and a half.
Put them to sleep.
Now they can listen to you again.
Most of the questions I get by email from the class are about TWiV episode this or that.
It's good. Well, they should send them in if you can't answer them.
Yeah. I
did send one in, yeah.
They want to know what Alan meant in some episode, and they give the exact second at which he said it.
Oh, cool.
I'll send it off to Alan. We have second paper today, and I thought this would be cool because this is a virology paper published January 2012. In vitro evolution of H5N1 avian influenza virus toward human-type receptor specificity. And this is a study partially done in ferrets, which escaped the claws of the NSABB.
This never should have been published.
And so let's just review H5N1 influenza a bit. This is, of course, an avian virus that emerged a very pathogenic form emerged in the late 90s, in the 90s, jumped into people in the late 90s in Hong Kong, and now and then infects people. It's gone back into wild birds. It infects poultry and causes losses. And the worry is, of course, that at some point it will evolve to transmit among people, which it does not do at the moment. And a lot of people are trying to look at what controls transmissibility. And in this paper, they adapted the virus, a wild isolate from Vietnam, to change receptor specificity. So birds predominantly have alpha-2,3 linked sialic acids, which are receptors for this H5N1. It does not particularly like alpha alpha-2,6 linked sialic acids. Those numbers refer to the sugar, the first sugar on a glycoprotein chain, sialic acid, how it's linked to the next one, either by an alpha-2,3 or alpha-2,6 carbon-carbon linkage. So human strains like alpha-2,6, our lungs, our upper tracts have alpha-2,6 sialic acids. Our lower tracts do have alpha-2,3, and it's thought that you have to really inhale these avian strains from an infected animal deep into your lung to get infected.
A bunch of it.
Yeah, quite a bit. So they did a really cool thing here. They put alpha-6 linked sialic acids on magnetic beads. And then they took a stock of H5N1 influenza virus, and they mixed it with the beads, and then they use a magnet to pull out the beads and whatever virus is stuck. So they're looking for a minority virions that can bind alpha-2,6, and they do a couple of cycles. They amplify after each. They elute the virus, they amplify, and they do it 4 times. And they get about a dozen viruses that have started to go, well, they bind the beads, let's put it that way. And the mutations are largely in the hemagglutinin in the pocket where sialic acid is known to bind, and there are a few mutations elsewhere
now,
on
this
the
is
HA.
an interesting feature of this is that they didn't have to mutagenize this virus or anything.
Right.
Because these are the spontaneous mutation rate in these RNA viruses is quite high. So these are mutations that are pre-existing spontaneously in a population of viruses.
Right, and in fact, I think they mentioned maybe in the introduction that that isolated strains from clinical cases of H5N1, the people who have actually shown up at the hospital, they may actually have some preference.
Right.
Or maybe in the process of developing some preference for this. So if you put pressure on the virus to adapt to human sialoglycans, it will.
This is an interesting point. So, as you isolate these H5N1s from the sick people and test them in the lab, they seem to be trending towards, they're evolving towards alpha-2,6 specificity. They still maintain alpha-2,3. And so the question is, how is that happening? Is it the pressure of infecting people? I mean, that would suggest that there's a lot of infections going on, because just one sick person, it's not going to evolve to alpha-2,6.
Or it could be that the people are panning for these viruses the same way you're doing with magnetic beads, right? The viruses that manage to infect the people are the people who have those, are the viruses that have that receptor specificity.
Right.
Yeah.
There is another possibility, and that is that certain wild birds have alpha-2,6 sialic acids in their gut tracts. In birds, this is a gut infection. Mm-hmm. Passes through the GI tract. So that could be enriching as well. It may have nothing to do with people, or it could be both.
Right.
So they do a couple of cool sugar assays. One is they do an ELISA where they put different sialic acids on plastic, and then they bind virus, or they put virus on plastic and they bind different sugars, alpha-2,3 or alpha-2,6. And they basically show that some of these mutations do, in fact, enhance binding of virions to alpha-2,6 linked sialic acids. A couple of them also decrease alpha-2,3 binding. And that's interesting because when pandemic strains emerge in people that seem to have come from bird strains, you have a loss of alpha-2,3 binding and a gain of alpha-2,6. So that's, they're sort of looking for this phenotype.
Right.
They also do a sugar microarray where they put all kinds of either 2,3 or 2,6 length sugars on a slide. It's sort of like a microarray, except instead of using nucleic acids, you use sugars, and you can get an idea of how these viruses are binding.
I thought that was really cool.
It's a really neat assay. You get a lot of data from it. And basically, the individual mutations, some of them have minimal effects on increasing alpha-2,6 binding, but when you start combining them, you put two together, then you get more striking acquisition of alpha-2,6 binding. But you never lose the alpha-2,3.
Right.
And then they take a wild isolate, which is from Egypt, from an egret, which has a couple of mutations that have made it more likely to bind alpha-2,6, increases its alpha-2,6 specificity. It has 2 mutations in the pocket, the HA pocket. And they combine those with some of the mutations that they have selected, and they get one virus that has 3 amino acid changes in the hemagglutinin, and it's basically lost all alpha-2,3 binding and now just binds alpha-2,6 sialic acid. So they've been able to. So it's interesting. By selection, they weren't able to do this, but by combining with naturally occurring mutations, they did. And then they stick these in ferrets.
You mean literally?
Yeah, they put them in the nose of ferrets. As you know, Dickson, ferrets are a good model for flu.
They're wonderful, but they're not people.
They are not people. I got to take a side step here. There's a letter in the Times on Thursday from the director of biosecurity at the University of Pittsburgh. And he wrote, because ferrets are a good model for influenza transmission, we therefore assume that these ferret-passaged viruses will transmit among people. I would fail a first-year graduate student for saying that.
Yeah.
Even a second or third or fourth year. I'd fail you, Dickson.
Even I would never say that.
Are you guys stunned by that, or is it just PR?
Well, look at. I hate to question people's motivations, but I think you have to in a case where somebody's paycheck is coming from this whole biodefense thing.
Right.
And here's somebody who's part of that coming and saying, "Be afraid."
Yeah.
And it's just the science absolutely does not support that.
All right, so on to the ferret experiments. You put the virus in the nose of a ferret, and if you just take this egret virus, remember it had a couple of amino acid changes that allows it to use alpha-2,6 but still does alpha-2,3. These viruses multiply in the ferret nasal tract, but they don't move to other ferrets either by direct contact or through the air.
So I assume the way they set this up is you stick an infected ferret in a cage with other ferrets and next to a cage with other uninfected ferrets.
Precisely.
And they transmit within the cage but not the other cage. Then the direct contact did it. If they transmit between cages, then it was aerosol.
Let me, by contrast, tell you a story briefly about the transmission of West Nile virus, which we know is transmitted by mosquitoes. But if you infect crows, which is common in nature, and you put an infected crow with a bunch of uninfected crows in a small container, they all become infected. Mm-hmm. So that's an easily transmitted...
In the same cage.
In the same cage by nasal secretions. So that's a virus that absolutely does that. So that's...
What if you put them in separate cages?
That's a good question. I don't know the results from that. I don't know.
Okay.
But I know at Cornell they did the other one and got it to transmit.
By the way, these ferret experiments are done at BSL-3 AG conditions.
All right.
It's not easy to do. You have to be careful.
So the AG, what does that say?
So that's the Department of Agriculture, USDA, specified above BSL-3 precautions.
So it's kind of BSL-3
plus for H5,
Rinderpest free.
Right, so that's not the bubble suit lab you see in the movies, but it's close.
It's the next step down.
Everybody's gowned up and masked and airlocked in.
By the way, one thing we didn't discuss when we were discussing contagion last time was that I thought that the representation of the BSL-4 lab and the BSL-3 lab, I thought that was done well. Not that I've ever been in one, but I think it was probably pretty accurate. It was pretty good.
I have a former student, Julie Harris, who works at the CDC, and she said they came to the CDC and talked to them about how stuff works, and she said they got it pretty well.
Good.
They asked her what do you wear on your feet, and she said whatever's comfortable. All right, the next virus is the combination of the egret changes plus a couple of the ones they selected for, and this infects ferrets. It transmits by direct contact, but not by aerosol. Okay, so improving the H alpha-2,6 interaction gets you contact spread, but not by aerosol. Then they did something which is really very clever. So they have adapted the H5 to do better with alpha-2,6 linked sialic acids, but a key protein in flu which allows the virus to spread from the cell is the neuraminidase. So when the viruses bud off the surface, they stick to receptors unless the neuraminidase cleaves off the receptors, the sialic acids, from the cell surface. And so the sialic acids fit into a pocket in the neuraminidase. So you can imagine that there is also specificity as far as what the NA recognizes. So they said even though this virus recognizes alpha-2,6 by the HA, maybe the NA isn't very good at cleaving it off. So they put in a human N2 neuraminidase from an H3N2 strain into this virus, which already has the amino acid changes that allow it to bind alpha-2,6, and that one is it's transmitted by contact in 1 out of 2 ferrets by aerosol. It's not a lot. These are small numbers.
It's a small N.
Yeah, two ferrets for each experiment. So, but then they tried an H1N1 neuraminidase, and that does not travel by aerosol. So even though both neuraminases are good at cleaving off alpha-2,6 linked sialic acids, they don't work in the context of this H5 virus.
So you need multiple changes. You need the HA, the H and the N participating in this,
right?
And not just any combination of those two will do. So it turns out to be fairly difficult to get a transmissible ferret virus.
Right. In fact, their last sentence is, these results show that considerable functional evolution is still required for acquisition of transmissibility in humans. Or they suggest this would indicate, because we don't know what this means for people.
They don't comment on its pathogenicity.
Yes, that interests me.
I suppose it wasn't, right?
Yeah, I mean they're doing nasal swabs on these ferrets, so I guess they're still alive.
I mean, it was my impression that H5N1 was lethal, but wasn't that what happened with the Fouché experiments? Well,
yeah, I think you started out with a lethal virus.
My last reading of the Fouché paper,
right.
Yes.
Are there any statistics on that table at all?
No.
That seems kind of one.
There's only 2 animals in each case.
Statistics wouldn't really tell you anything.
I'm wondering if that one animal could be an anomaly of some kind too.
Absolutely, sure.
I suppose another thing. So I lost track here. So in the one that worked, the mutants in that virus included the ones they selected with beads, right?
Yeah.
Yes.
Yeah.
It's sort of a combination of the naturally occurring mutations and the ones that they identified with their panning with beads.
Right.
So what's interesting too is that it's also not clear whether that combination would ever arise in nature, because they weren't selected by viral replication; they were selected physically.
Absolutely.
Right. So they may be things that they appear and go away immediately in an infected individual.
I mean, this is in part why there's fear about the Fouché virus, because they say he actually selected for viral replication and transmission in ferrets. So it's worth reviewing what he did. So Fouché introduced 5 amino acid changes into the HA, which changed the specificity to alpha-2,6. And then he infected ferrets with that, took a nasal wash, infected another one, did that 10 times, and then supposedly by the end, these things could pass by air from ferret to ferret. I don't know where the mutations are that do that.
And we don't know if that's reproducible.
We don't know anything, because he's the only guy who's saying it.
And Kawaoka came out and did finally say something to the media. He's been silent so far, but it sounds like his experiment yielded a rather different result.
Yeah, so what Kawaoka did...
Well, it's a different experiment.
It's a different experiment, too, yes.
So I assume Kawaoka put the same alpha-2,6 mutations in, right, into HA. I'm sorry, no, he took... I'm going to start over.
The whole gene.
Kawaoka took an H5 and put it into a H1N1 background.
Right.
And that transmits but doesn't kill the ferrets, right?
Right.
That's right.
Now, I don't know if that H5 was also changed to allow it to recognize alpha-2,6 or not.
Right. We can't really go into these because nobody's seen them.
But his was not lethal.
Actually, probably hundreds of people have seen them, but we haven't.
But
to
Alan,
me, the interesting thing about that is that it says that gene segments other than the H5 affect transmission.
Where does that say? Where?
If you just Kawaoka's experiments, if you take the H5 hemagglutinin, the H5 virus doesn't transmit, right?
Right.
And if you take that hemagglutinin and stick it in a virus that has gene segments from the normal pandemic, now you get something that's transmissible. That says that gene segments other than the hemagglutinin impact on transmissibility. So that's it. I would predict, therefore, that if we ever see the paper, that if you look at Fouché's, you'd find that some of the mutations that affect transmissibility are in genes other than the HA.
Yeah,
okay?
That's
but that could be anything, because just it could be a numbers game. It just could be that the amount of replication or whatever could affect the transmissibility.
Sure.
So, don't know.
So
don't know.
here again, from the outside looking in, I'm a virus, and I'm trying to get into this host, and I can't get into the host. So I sit there in front of the safe, that's the host, and I keep twisting the tumblers, and I get the goddamn door to open. But wait a minute, there's another safe inside. So this is like 5 safes. This is like a Russian doll situation.
Matryoshka, is that what
Yeah,
it's
the
called?
first door opens, and oh my God, I have another safe to open now. And by the time that happens in nature, it's probably going to burn out.
Well, if this were easy for H5
Forget
to do, we
about
would have
it.
seen an H5 pandemic by now.
Forget about it.
Right, we've seen other flu viruses jump to humans in the past.
Of course.
And the question is, what would it take for an H5 virus to do that? And at least from the published data, it looks like it would be kind of a tough trick.
Tell me about it.
Can I ask a naive question?
Please.
So you gentlemen have followed this a lot more close. I mean, I've been aware of it, but I haven't followed it closely. I'm not a flu virologist. I'm a virologist, but I don't work with flu, so I only know what I learned in textbooks. But it's like in all this hubbub, without even seeing the paper, I'm guessing that this isn't some rare and brilliant insight that led to these experiments. I bet I can guess what they did.
Now, this is
And I bet anybody with the
force.
technical know-how to reproduce this.
Of course.
Right.
So
Well,
why?
So by drawing attention to it, you've already let the cat out of the bag.
Yeah,
right?
from the published
Is it cat?
descriptions of what was done, it's actually all you would need to reproduce these experiments would be a load of ferrets and some H5 isolate.
The same.
But even without those descriptions, right? I'm sure anybody working with flu has at times thought about these very ideas.
Sure.
But of course.
Right? If you're a virologist, you're always thinking about things like how do you change receptors and how do you target different hosts. So there's nothing. This isn't some brilliant secret that had to be locked away. Somebody else would have done it if these guys hadn't.
I mean, HIV has to jump over 3 hurdles in order to escape from all of the triple treatments that are now in place, right? The hard treatments. There are 3 blocks that are set up among the drugs that are used. So mutants that can go around 1 can't go around the second. They can go around the second. They can't go around the third. It hasn't happened yet, or has it?
Yeah, but I think
the question is,
do
we
even
have
if
a
it
triple
happens-
therapy mutant of HIV that no longer responds to triple therapy?
I don't know.
I think you do get. I
It's
think
a
you do
crippled-
wind up getting resistance
crippled
out the other
virus.
end, and then you just change the cocktail.
Okay. So
Yeah,
how
it's
long
a-
does that
virus is
take to
probably
jump
not
over
a-
3 hurdles? That's what I'm driving at. And that's
Well,
a-
and what
not-
does it what does it cost is the other issue.
Yeah,
because
yeah.
the virus, my understanding is, as HIV evolves resistance to these drugs, it's not that the virus is hale and hearty and resistant to
Right.
AZT. It's a virus that's barely kind of limping along and resistant, and then it hits this. It's another chemical.
Yeah, exactly.
It hooked its toe on one of the hurdles and fell and broke it.
Very good.
That's right.
We could take this.
Capsule has a hole in it now.
So Welkin, Bob Lamb has said the cat is out of the bag and it's a ferret. So Welkin, although you've followed this story, what are your thoughts? Should we worry about this? Should we publish it? These ferret results?
I think so. I because I think that's what I was getting at. That I don't think you're not going to be giving anybody who could reproduce this for nefarious reasons will already have thought of these ideas or could easily think of them anyway. But the flip side, I don't know if you guys saw, but Kawoka did, I think, just published a comment in Nature a day or two ago.
Yeah.
Mm-hmm.
And sort of, he also just makes the argument that viruses like this could become a problem, right? And you don't want to impede research.
Exactly.
Right.
We should be prepared and understand how these things work.
Exactly. Exactly.
I want to be careful what I say.
I mean, I think if they were done in
I'm not tenured yet. I have.
if
you
they
guys
were,
can say
if
whatever
they
you
were
want.
done in a BCL4 or BCL3, a
BSL.
BSL, I meant BSL, biosafety.
Well, some of
laboratory.
these people are insisting that they be done in BSL-4, and that's the ones in none of these have been done in that, and you don't have those everywhere.
No,
so
you don't. No, you
that's
don't.
effectively stopping the work for the most part.
I understand.
I don't think it requires that. There was an article last week in the Times by Denise Grady, I think, which was equally messed up in saying that there's 50 to 60% fatality rate. But she said the scientific community is divided over this issue. And someone wrote a comment on Virology blog and said, is it true? Are scientists divided? And personally, everyone I talk to, and it may be just the people I talk to, are fully in support of publishing this. I signed a letter with 17 other virologists who said it should be published. As far as I can tell, there's Rob Webster and then the bioterror complex that doesn't want it to be published, and most scientists seem to be behind this.
Yeah, that's true. Vince, please let me
Thoughts?
read.
Hold on.
Let me read the introduction to this paper.
Go ahead.
Circulation of H5N1 highly pathogenic avian influenza virus in Asia, Europe, and Africa has resulted in more than 550 human infections since 2003 with now I'm going to underline this, put it in red, and let you read it, with a case fatality ratio. Has anyone in the common press ever called it a case fatality ratio? And the answer is no. And what is a case fatality ratio? Notice that one of the authors is from the Centers for Disease Control. They say case fatality ratio because the number of people who get sick versus the number of people who die, that's the case fatality ratio. That's 58%. But then they go on to say, this number of human cases is just a minute fraction of the large number of people likely exposed to H5N1 during thousands of H5N1 outbreaks reported in poultry, indicating that virus transmission from birds to humans is very inefficient. So when they start to quote these big numbers of deaths, they're only talking about the people who were admitted to the hospital because they were sick. It's unfair because if you really want to know what the fatality rate from this thing is, it's probably less than 2%.
Well, they don't even mention that there are probably a lot of inapparent infections based on serological surveys. Okay,
well,
so they're not doing justice to the data in this paper either. I don't know why people do this.
They should say what the death rate is from the number of infections versus the number of deaths.
Well, we don't know the denominator. That's the problem.
Well,
so
but they say it's big. Even they say it's big.
No, they say exposure. They don't say infections. The human case is a fraction of people likely to be exposed. They argue that transmission from bird to human is inefficient, and I would disagree based on the serology. I don't think that's true.
Yeah, you don't develop antibodies against every virus that you ever smell.
No, that's correct.
You develop antibodies against viruses that your immune system thought were a threat, and it probably thought that because they were replicating.
But I just wanted the case fatality ratio explained.
Yeah,
because that's an important number to keep in mind. It's not a
and a good example of this would be cytomegalovirus, right?
Yeah,
a good chance, probably at least 2 or 3 of us who are talking right now have this virus.
Sure,
and it'll never be a problem for us.
Nope.
But if you look at cases, well, it can cause serious problems.
Sure,
but that's kind of a freak accident for the host and the virus. The infection wasn't supposed to go that way.
Right.
And I think H5N1 is probably going to turn out to be the same way when you look at the seroprevalence and you say, well, probably millions of people have been infected with this thing. It didn't get anywhere because we're a crummy host for it.
All right, so that's the paper that was published. There you go. We'll keep you up on this.
Hopefully more papers will be published on this sometime soon.
Watch this space.
We should point out that there has been a voluntary moratorium on H5N1 transmission research.
Yeah, which was about as voluntary as my tax return, I think.
So,
during that time there will be some.
But that's only in this country.
Discussion.
No, it's global.
It is global.
Yeah. All right. So they're going to have a discussion and decide how to go forward. So we'll see. I think it's unfortunate to stop. These data are interesting, and it would be nice to know what the others found, and we could talk about it and have a nice discussion. But
and also, despite talking about the seroprevalence data and saying this virus is probably not the kind of threat that it's been made out to be in the past couple of weeks, it is nonetheless a flu virus. It's circulating in nature. It's capable of going into people. It's for the 600 people who did manage to get hospitalized with it. It obviously was a serious problem.
Yeah,
and we need to know more about that.
Yeah, but wait a minute, the other group lived.
Yes.
So not everybody dies from this thing.
Right, but exactly. And we need to know what's going on with those people who got sick with it and what's causing some of them to die.
Here.
And also, is this something that could, even if this particular virus can't make the leap into humans, is it something that could potentially exchange genomic information with other flu viruses in a human case and be a source for new pandemics?
Right.
So it's an interesting area that needs more work.
We should point out that Palazzi and Wong had an interesting PNAS paper this week. I don't know if you guys have seen it, but
I've seen it. I think that's really a great write-up.
Yeah, it's a summary
really
of
good.
Why this is all ridiculous, and we'll put a link to that in the show notes.
That's a really nice piece of work.
Let's do some email. The first one is from Mike. Hello, men and sometimes women of TWiV.
I'm never a woman. What does he mean?
That's right. Very good.
Not even some of the time.
Years since I wore a dress.
Alan.
I have read before that the human genome contains the genetic code of several thousand retroviruses. These retroviruses are in an inactive state and are believed to be the product of infections that have been passed down through the germline through many generations of humanity. These viral genomes have, in effect, become incorporated into the human genome and are now a part of us. I'm wondering if it would be somehow possible to use some form of restriction enzyme therapy to remove these viral genomes in order to see what effect, if any, it would have on human health. Do you think it could be done? If so, what would you predict the effects would be? Are the viruses in any way, shape, or form driving evolution, or could they be causing damage just by being there? Once again, I appreciate you taking the time to read this email. I greatly enjoy listening to your podcast, and I apologize if this is an ignorant line of questioning. I started listening to your podcast regularly in November of this year and have enjoyed it so much I'm now trying to catch up. I'm currently still in the early 2009, but should be completely up to date within the next 2 months or so. Once again, thank you for all that you do. P.S. Please tell Alan that I have already taken the listener survey, so if he could please remove the subliminal message from my brain, it would be greatly appreciated.
It's got an earworm.
I would very much like to stop waking up in the middle of the night with the sudden, inexplicable urge to take a survey over the interweb.
Mike, this is a subliminal message. You no longer have to take a survey. Simply write your check and... Oh, sorry.
Here you go, Welkin. That's for you.
This is for me? Oh, okay. No.
Okay, thank you very much.
Next letter.
No, letter to follow.
Next email.
That's
so
right.
I don't think restriction enzyme therapy. If the idea is, could you cut every single one of these out of the genome and stitch the genome back together? I think the answer is no. I don't think that would be at all possible. There's hundreds of thousands of them in the genome. And of course, you'd have to knock them out of every cell, right? You'd have to treat every cell. But I think the other parts of his questions are pretty interesting.
Or just the germ cells, and then make an embryo from that.
Yeah. Yeah, I suppose engineer, but even that would be impossible. I don't think there'd be any way to do that. And I don't think you need to. I don't think they're an issue. The one piece of information I found, he reminded me of an old paper. I forgot to write down the name of the authors, but there was work in the chicken system back in the 70s and 80s where they had some of these elements were polymorphic. I'm sorry?
Sorry, Astrin, Bus, and Haywards.
Yes.
That's right, Astrin. They had chickens that were polymorphic for different endogenous loci, meaning that some chromosomes had them and some didn't. So they were able to crossbreed these and breed chickens that were lacking the particular family of endogenous viruses that they were interested in. And the fact that they were able to breed those and grow them, they concluded that meant at least those particular herbs were not required for the chicken to be viable and getting rid of them. But it's an interesting experiment. It was published in Nature, but it doesn't really tell you much because there was probably still tens of thousands of those things in the chicken genome other than the ones that they bred out. Yeah. So that's the closest thing I could find. The rest of the other questions are very interesting, like the thought experiment of what would the effects be. I've always assumed that there's so many of them there that they probably really have become part of the architecture of the genome in different ways, and that spontaneously getting rid of them probably would have huge deleterious effects on the genome in some way, the way it's replicated or the way it's packaged on nucleosomes or something. Yeah. They contribute now to the spacing between genes. They make up parts of introns. They've essentially become part of the genome. And actually, maybe the way to think about this. So his third question, are the viruses in any way, shape, or form driving evolution? It's important to remember that these sequences, they're not viruses. They were viruses. A virus put the sequence there, but the sequence itself is no longer a virus. It's just a string of nucleotides, right? It's just DNA sequence.
Right.
Yep.
And most of them in the human genome, as far as we know, aren't even capable of making virus anymore.
Welkin, do we have any data from the Neanderthal project on this?
Boy, I should know that. I can't remember, but I would bet good money that Neanderthals have almost all the same ones that humans do.
I'd be shocked if it were much otherwise.
Right, because we share quite a few of them with chimpanzees, for example. Right, and all there's a huge subset of them that all humans have in common. So it's certainly the case that we have them in common with Neanderthals.
So how far back would you want to go on this?
How far back would I want to go?
In evolutionary history, before you start to say there aren't hundreds of thousands, now there are just hundreds, or maybe there are 10.
I think the retroviruses are so old, right? So Rob Gifford and his collaborators, for example, have identified some of these that are tens or even hundreds of millions of years old, wow. Right? Even though they're highly degraded. The retroviruses have been around for so long that I think that we were probably at an equilibrium hundreds of millions of years ago.
Wow.
In other words, I don't think we steadily accumulate more of them. We probably lose them just as fast as we acquire them.
Got it.
Right? Because just like any other sequence, any other non-functional DNA sequence, over time, if it's not doing anything, it gets spit out and that spreads through the population. But at the same time, you're occasionally acquiring new ones.
Sure.
Although at this
I
moment,
have nothing to back that up. That's just my guess.
At this moment, there could be someone on Earth being endogenized by a lentivirus, and in 3,000 or 4,000 years, we'll see the result, right? It's about,
sure.
Didn't we have a show on that once also?
Yeah, it's Welkin's first show.
Right. Gee.
It's okay. It doesn't matter. It's over a year ago.
How quickly they forget.
Yeah.
So there are so many primates.
New listener, Dickson.
There are so many primates. In other words, all of those primates would probably share the same batch of retros. All of them.
Or very similar.
All of them.
Yeah, it's
Okay.
You can classify them by how old they are, right? So there are ones that we share with chimps that we don't share with orangutans, for example.
Right.
So
that's
that means those viruses were around much more recently. That would have been an epidemic that happened 5 or 6 million years ago.
Yeah, and you can only go a certain distance back before they would become unrecognizable, right?
Yep.
Yeah.
Because you'd accumulate mutations to the point that you can't really say that's a virus anymore. It just
All right,
thank you. Wait a minute, I want to tell Mike, go listen to TWiV 108 on the tree recombinase, and TWiV 144 on the zinc finger nuclease.
Yes,
both techniques for using sort of, as he said, restriction enzyme therapy, some sort of enzyme therapy to engineer the human genome. It's not a crazy idea. People are thinking about it, and to some extent, they're doing it.
But don't cut out your retroviruses. Right.
But for what he's asking, what you would be talking about here would be engineering a new human being that didn't have
that's the problem.
Right?
Yeah, right.
To actually rid them from a person completely, you'd have to hit every single cell in the body.
Yeah.
Right.
Yeah.
Right.
Alan, would you read the next one, please?
Sure. Rehan writes, "Hello, TWiV hosts. In TWiV 167, Dr. Despommier said the virus in Contagion is similar to Nipah virus, as the virus in the movie is erroneously compared to a bat-pig hybrid. Dr. Despommier said about Nipah as, quote, pigs and bats gathered together in the mangrove. Nipah viruses arose in Malaysia, where pigs in a farm were feeding off bat droppings. The bats, in turn, were habitating in nearby fruit orchards. Right. No one would place a pig farm or fruit orchard in a mangrove." I guess I'm being as nitpicking as Dr. Racaniello was on TWiV 167.
Did you say mangrove or
Mango Grove.
Mango Grove.
That's what you meant, right?
Yeah, of course.
Right.
I didn't mean the mangrove.
The pig farms would be in a pig grove.
I never meant the mangroves. No. If I said that, of course, I take it back, of course. No, I meant a grove of mangoes.
Yeah, you've said that before.
Sure you did.
Grove.
Sure you did.
Right. Well, in the movie, they've
Nice
got a bat. They've got a fruit bat flying off what looks like a mango tree, and then
Right. No,
That's
that's
right,
exactly right. That's exactly right.
All
That is what I meant to say. I misspoke.
I see. He takes nitpicking from me, Dickson.
No, that's good though. I think that's
fine.
excellent.
They're listening.
Speaking of mangroves, have any of you guys ever been to the Okefenokee Swamp?
Yes.
Fantastic.
Yes.
That's
Amazing
a real
place.
It's fantastic. Is right.
I think I was, but I don't remember.
No,
when.
it's marvelous. Fakahachi is also
real?
very good down there, by the way.
excuse me.
There's,
No,
there's one over by Naples
family
that's also
show,
very
Dickson.
good. No, it's called a Fakahachi.
All right, we have time for one more. Rich, please.
Sure. Stephanie. Dear TWiVers, Stephanie writes, Dear TWiVers, I was reading a paper on exploring vector immunoprophylaxis for HIV and was surprised to hear that the transmission of HIV has been found to be less than one infection per 100 heterosexual coital act. When I looked up the reference paper, I found a control in the methods section so amusing that I had to share it with you guys. So basically, the transmission statistic was based on couples in Uganda with only one spouse positive for HIV. 80.4% of the couples reported never using condoms. The point of the study was to follow these couples monitoring HIV status to evaluate the transmission rate of the virus. After the study was over, they compared the strain of HIV from newly infected spouses with the strain collected from the spouse. Apparently, the authors had to exclude 4 people from the study because their strain of HIV could not have come from their partner. I wonder how each of the 72 couples that underwent seroconversion during the study responded when they saw that in the paper.
Wow.
She gives the reference for the paper.
The subjects were, of course, anonymous.
Had to happen.
Indeed.
They didn't even know about it until they heard it on TWiV.
Right. There you go.
This the paper, of course, she was reading was the one we did today, vector immunoprophylaxis. All right, I think we need to go on because Sophie's is up again, and that will take a while.
We really need to get to that
We
one.
really need to get to it.
Yeah, but it will take us a half hour.
You're right.
Yes.
So I'm sorry, Sophie. Next time, who's buzzing? Must be Rich.
Think so?
Well,
I don't start that till after 5 o'clock.
maybe
I
it's
don't think
not.
it's me.
How about Welkin? Are you buzzing? Unplug your headset.
Unplug it and plug it back in.
Yeah. There you go.
The buzzing stopped.
Uh-huh.
All right.
Was that me?
Yeah, that was you. It's after about an hour, the chipsets in those headsets start to go crazy. That's what I've been told. Let's do
The
a couple
chip in my head or in the...
in your head. Yeah.
Yes.
Yeah. That's it.
Let's do some picks of the week. Let's start with Welkin.
Well, I had one humorous one and one topical one, and I think maybe I'll go with the humorous one if it hasn't been done before, unless anybody has feelings on the
Go
matter.
for it.
Oh, do it.
Go for it.
This is a piece of correspondence that appeared in Nature a couple of years in April of 2004. It's called Irrelevance, Buzzwords, and Coffee. It's the virtual PI. And it's basically, sir, I'll read a little bit of it. Sir, several graduate students would like to suggest a modest proposal to the authors of the recent article on functional genomic hypothesis generation and experimentation by a robot scientist. And they refer back to an earlier paper. We've been doing a little research of our own and have generated an exciting hypothesis. We can build a virtual principal investigator. And this, I'll just read the next sentence. The software platform will consist of a tiny amount of background knowledge about a completely different system from the one actively being investigated, a buzzword invention engine, coffee consumption apparatus, a grant writing node, and a cat o' nine tails that will tie everything together.
Superb.
Great.
This is good.
And it's from, actually can't find the signature. It just says several non-robot scientists names supplied.
Right.
I think it was a group at Harvard Medical School.
Oh, that's funny.
Probably in somebody's lab there.
Nice. That's great.
It is.
I love it.
I like that. Good.
Very good. Thank you. Rich, what do you have for us?
I recently got an old 1999 Nova episode through Netflix called To the Moon. And it's a really nice summary of the whole manned moon project that NASA did, and I grew up with the moon shot, right? And so I love it. And this is a really nice summary of the whole thing. There's actually, the link I'm giving is actually to a website that actually gives you a link to the video, but also has some other stuff about that particular project. So I just thought it was if anybody's interested in the manned missions to the moon, this is a really nice summary. You can get it on Netflix.
As a follow-up, Newt Gingrich actually said something yesterday to the Florida public about wanting to establish a colony on the moon.
Yeah.
Did he say, to the moon?
No, he never said that one.
Oh.
Alice.
He'd make a good Ralph Cramp.
Yeah, that's right. You have to preface it.
Alan.
I have a pair of picks on a related one topic. So this is 2012. It is the 100th anniversary of the Terra Nova expedition to the Antarctic.
Oh, yeah.
And that was 1912 was the year that a team of British and a team of Norwegian explorers went and were the first people at the South Pole. And the British actually lost the race to the pole, and a number of them died on the way back, but they have a much more interesting tale to tell as a result. The Norwegian expedition, they went to the pole, they put their flag there, they came back, everything was fine.
Big deal.
The Robert Scott was the commander in charge of the Terra Nova expedition. That was the British effort. Amundsen was the Norwegian. And the Scott expedition was a scientific expedition as well as a put the British flag on the pole expedition. So in honor of the 100th anniversary of this, there is now, somebody has set up an account on Twitter. It's cool. Captain R.F. Scott. And he's tweeting. He's tweeting entries from the journal that they kept on their journey. And right now he's, I think, following the actual schedule of the march that they made back. So January 27th, 1912 is I think the posts that are coming up now. Spoiler alert, this feed is going to end in about March, late March. But the other thing is some photos from the expedition. That was the first scientific expedition to use photography. So there are some great old black and white photos of the guys trudging across the ice, and they some of these have been put online. So I'm giving a link to those as well.
Nice, cool.
For those of you not something I would like to do.
No, and it does not end well.
It looks really unpleasant.
It certainly doesn't.
Yeah.
I think the tweeting is great.
Yeah, I think
it's really funny.
The photos are terrific.
Cool. Dickson, do you have a pick?
I do have. I want to follow up on Alan's just a moment. There's a footnote to this, though. A woman soloing just skied across the entire continent, south of Antarctica.
Cool.
That's remarkable.
I wouldn't want to do that either.
That is remarkable. No. That's a little bit foolhardy, but congratulations to her. Yes. My pick is the Discovery Channel show called "Removing the Oceans." I don't think that's the exact title, but it's a program. Maybe it's sponsored by the National Geographic. I'll get the title of this right so you can put them in there. But it's a program that they show you what the bottom of the ocean looks like and how the continents moved around and where the geological faults are and all of the tectonic plate movements and everything. It's fascinating. What would it look like, for instance, Rich, if you could just go out to the Florida coast and then take all of the ocean away and look across at, let's say, the Bahamas, for instance. Cool. You stand on the edge of these islands, and there's just a cliff that goes down 4,000 feet off the one that I go bone fishing in, Andros, and that's called cliff diving. My God, when you look at it with all the ocean missing, you realize how remarkable the Earth really is.
Dickson, if you actually did that, what would happen?
You'd fall.
No, if you removed all the oceans.
Well, everything would die.
Exactly. All those viruses would be gone.
They'd be gone.
It's like removing all the endogenous retroviruses.
Well, I can tell you right now that you're looking at Mars with their oceans removed.
All
right, so Mars did have oceans at one time.
That's right. Thank you, Dickson.
You're welcome.
Alan is searching for it, I can see.
Yeah, I'm not finding
it's got to be there.
It's
got to be there. No, it's probably not there like that, but it's a program about removing the oceans from them. I'll get it straight.
I'm sure you will.
It was not an accurate pick, but it was a good one.
Maybe it exists.
See, if they show that show now, the terrorists will go and drain the ocean.
Well, now they'll know how. Yeah.
Oh, that's funny.
Remove the oceans.
Yeah, I think they're welcome. That's what they'll do.
My pick is a column by Robert Cringely. It's called I Cringely, his blog. He writes about technology, but I thought this was an interesting post. It's called Hello, Mr. Chips. And it's basically the idea that he thinks technology like iPads and so forth in classrooms is not really going to revolutionize education yet because we're not ready for it. And he got an email from a high school teacher who said basically kids who want to learn will, and those who don't won't, no matter what you give to them. And I was just wondering if some of our listeners who are involved in high school teaching would read this and send us your thoughts about it, what you think about this. He also writes that it's going to be about 10 years before the technology is really ready, and by then the current generation of kids who are into technology will be running the schools, and then it'll all happen. A decade from now, technology will be cheaper, and the lubricity of acquiring knowledge will be dramatically improved. I think time and space will cease to be factors in the educational experience. So yeah, go ahead.
I'm sorry.
No.
I wanted to make a comment on this.
Yeah.
A special case that I'm personally aware of is the iPads are turning out to be a real boon for autistic children.
Really?
Right. So kids who have trouble or can't learn to communicate with their voices, very often what they do is they communicate with picture cards. And the iPad, if you think, they've developed programs now, the iPad is so much more efficient than carrying around a notebook full of pictures, right? So they have programs on them now where the kid can touch the picture and even build whole sentences. Yeah. And when you think about it, an iPad, you could have millions of images on there. You're not restricted to the handful of the ones that the kids... So that's a very special case, but it's a... for the purposes of their education, I think it's going to turn out to be important. It's happening right now.
That's a good point.
Interesting.
That's a very good point.
Yeah, I found this post very interesting, though, because I think there probably are a lot of cases where schools are just saying, well, stick technology on it.
Yeah. Right.
Yeah.
That's not not the approach
right.
that really needs to be used.
I gave a talk at a Gateway school 2 days ago, and it's in New York City, and it was founded to take care of the need to teach special ed kids who were very bright but also disturbed, and disturbed in ways that they were probably bored as hell with their learning because they weren't learning the way they need to learn. And what they were employing there was play therapy. So they were building a Rube Goldberg apparatus, for instance, which had multiple functions and had to accomplish something at the end. But they worked as teams, and they understood the science behind it, and they were doing extremely well, working with their hands and their mind together. And they could use the internet if they wanted, but most of them chose to work hands-on on something that you gave them. So the trend nowadays in medical school, at least, not to say that those are disadvantaged children learning at a poor rate, is to take away that laboratory experience from them. We just did that here at Columbia University. They took all the microscopes away from the students. So they have no microscope learning experience whatsoever for the first 2 years of their life as young doctors. And I think that's crazy.
Yeah.
To be honest, I think it's crazy. Too bad.
Anyway, our teachers out there, check it out. Tell us what you think, because you're out there in the fields doing this. I mean, this is written in response to Apple's announcement last week, of course, new educational tools, and they're saying, the iPads are going to be good for everyone, but I just wonder what you think.
These kids, by the way, didn't have learning disabilities. The teachers had teaching disabilities, which brought them to that school.
A common problem.
Yeah, yes. No question about it.
We have a listener pick of the week from Charlotte, who writes, "Contagion, what a dog."
Oh, I don't know. Come on, that's a little harsh.
Well, you didn't see it, Dickson.
Yeah, but it's still harsh. Can't we ease this down a little bit?
Yeah, it's good, Charlotte. I met Charlotte at a meeting.
How funny.
May I recommend to my fellow TWiV addicts a movie about a real virus, Roger Spottiswood's film, and the band played on. based on the book by Randy Schiltz. I've never seen that one.
No.
Nope.
And we have never picked it, of course.
That's true.
I've seen
Have
it
you?
a few times.
Good?
Yeah. It's good. It's interesting to watch, especially if you're a virologist, because it's interesting because it's about the beginning of the AIDS epidemic in California.
Mm-hmm.
But it's also interesting because of the skew in the movie. So, for example, Alan Alda plays Bob Gallo. There's a scene with Alan Alda sitting in his office, playing Gallo, and he's like underlit, so he looks maximally evil, with shadows cast up on the ceiling and everything. So it's quite clear they had a particular point of view in that movie.
How funny.
But it's still an interesting one.
The Nobel Committee apparently agreed.
Yeah, maybe it influenced them.
I have to read the book first, then I will see the movie. I prefer to do that in that order. She also writes, and for my fellow TWiV addicts, Akira Kurosawa's 1952 classic Ikiru about Helicobacter pylori, although not explicitly stated in the film. Of course, it hadn't been discovered yet, right? Perhaps a listener pick when you bring Marty Blazer up from NYU. Looking forward to seeing you at ASM in June. Perhaps we can do a live show at Berkeley.
Ooh.
Charlotte
Berkeley.
is at Berkeley.
Right.
Okay, Charlotte, thank you very much. And thanks, everybody. That'll do it for TWiV 168. You can find us, of course, on iTunes and at the Zune Marketplace and at microbeworld.org. You can pick up our app for iPhones and Android devices over there as well. Do tell us your question. Do send us your questions and comments to TWiV at twiv.tv. We love to get them. Welkin Johnson is at Boston College now, and he sent his link showing him very handsomely sitting in his new laboratory. Look at that, with a jacket on. Thank you for joining us, Welkin.
No problem. I'd love to be here.
Yeah, it's a lot of fun. Come back more often.
Definitely.
In fact, you're on the email for the schedule, so whenever you have time, you are welcome.
Oh, great.
Dickson Despommier.
Yeah, Vincent Racaniello.
He's right here at Columbia. And verticalfarm.com, Racaniello.org. Wait, medicalecology.com.
Good.
Thank you, Dickson.
You're welcome. Pleasure to be here.
And as we were talking, the sun came out, the sky became blue.
That's right. And
now it's back to the other stuff.
Exactly.
I don't know.
It's back to the Robert Gallo for you.
Alan Dove is at alandove.com. Thank you, Alan.
Always a pleasure.
And you're back on the Twitter.
Yes, I'm back on Twitter again.
Alan Dove, all one word.
Yes.
There you go. Check it out. Rich Condit is at the University of Florida, Gainesville. Thank you, Rich, for joining us.
Quite welcome. Always a good time.
Oh, you've been quiet a bit here.
Oh, I speak up now and then.
I haven't heard you for 30 minutes, right?
I just listen to you guys. You guys
He was
are
in for
pretty
the AAV
entertaining.
discussion.
I'm kidding. Thank you, Rich. I am Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. Anyone listen to last week's episode past the music at the end?
Yes, I did.
I don't know if anyone ever listens.
Oh, they
fast.
do. Kathy said that's one of her favorite things.
Wait for an Easter egg, right?
Is those little the little bits at the end. That
All
we
right.
Sometimes put in.
At a movie, sometimes at the end when they're playing the credits, they show you the
yeah.
outtakes.
Sure, the outtakes.
Well, they
I
show
always
the
wait.
outtakes.
I wait because I want to see something, and my family's like, "Let's go, let's go."
Exactly.
No.
I do the same thing.
I
do
love
that.
to sit and watch.
Toy Story,
funny.
of course, was the best. They had all those outtakes. at the end.
They had actually, I think, some of the best outtakes. Did you see A Bug's Life?
Yes,
Yeah, that was great.
Oh, just terrific.
it's
was great.
fantastic. It's fantastic.
Where it was like the cartoon characters were messing up on set.
Exactly.
Yeah.
Yeah, that's right. That's right.
That's right. Cartoon
great.
characters doing outtakes. It was great stuff.
I think Monsters, Inc. I think actually all the Pixar films have done that.
Do that. That's right.
You know, I have all the recordings of TWiV since the beginning. There's so many outtakes in there. If someone wanted to go through and find them, they could make a whole show out.